Hello, my name is Felipe from Brants, and this is my presentation on the effect of stress on C. elegans and its progeny's likelihood to undergo dower formation. Epigenetics is the study of how the environment changes gene activity. The environment serves as a stimulus for adding and removing methyl and acetyl groups from the DNA and histones, changing the cell's response to the environment. In 1998, it was discovered that epigenetic factors can be passed along to the next generation, thus opening the field of transgenerational epigenetics. Further, in 2014, Diaz and Ressler discovered that this effect can be induced in mammals and opens the door for transgenerational epigenetics having an effect on human health. However, currently the studies that have been done on this field mostly see this effect as being random, and the goal is to see if a pattern can be found along stressing the parental generation and the filial generation, with my hypothesis being that there is a positive relationship between stressing the parental generation and the stress phenotype being more present in the filial generation, and that the HERDI1 pathway is responsible for the epigenetic inheritance that we see in our model organism. The research used C. elegans as a model organism because of its high use of argonauts, similar to humans, to control the RNAi interference. And in addition, C. elegans are really useful to our research because they eliminate the factor of genetic variation between the offspring, since they're all genetically identical. And they use the dower mechanism, which can be used to distinguish stress. When C. elegans are stressed at an early age, around the larval stage 2, they can choose to undergo an alternate cycle of life, which can be seen specifically through their different phenotype called the dower. The dower stage can be exploited for us to understand and see the effects of epigenetics faster by determining which ones are not stressed and stressed, based on which ones can eat, with dower organisms not being able to eat during the dower stage and later being able to eat when the stress is gone. So the experimental setup essentially is that we would take the stock generation and then separate it between stressed and not stressed groups. After we stress them, we would remove the stress or no stress of them to a different petri dish so that they can reproduce. From there, we take the offspring and then we do the same, but this time we add them into a petri dish with fluorescent beads. The ones that glow means that they were able to eat, and since they were able to eat, it means that they were not in the dower stage and were not stressed. So based on the different numbers of dowers per generation, we can see if this effect was passed along. The strains that we're going to be using of C. elegans are the N2, which is the most commonly used strain as an average strain, and the HERDI1 strain, which has a defective argonaut called WAG09. We're also going to be testing for two different types of stress, which are starvation and overcrowding. Overcrowding will be tested using pheromones so that we don't have the complication of starvation being a confounding factor. And finally, the statistical tests applied to compare the groups were the ANOVA test and the t-test. So we can see the data at the bottom here between the three graphs, and we also have a picture that I took from the microscope. And comparing the stressor trends and the pheromone trends, we can see that the stressing the parental generation for pheromone led to less dowers in comparison to the starvation, which led to more dowers. And as a result, we can see that these two different stresses have different mechanisms for which they follow, and thus prove my hypothesis wrong, in the sense that the suggests that there is different activation mechanisms between the two systems. Also comparing the different strains, when we stress the N2 strain, there was less dowers in F1 in comparison to the HERDI1, which had more dowers. So that suggests that the WAGO9 Argonaut is involved in the passing of the genes of the epigenetic traits further, thus supporting my hypothesis. And so overall, my research was partially supported by the data. So looking into the future, I would like to research further into how the Dower starvation dynamic works, because we were not able to test the starvation group for the Dower, for the HERD1, unfortunately, due to time constraints. And we would like to see if different Argonauts can have the same effect. The implications of this research is that it can give us a greater picture of how the genes interact with how the environment interacts with the genes, which can help us develop in the fields of hormones, cancer development, and cell differentiation. So thank you very much.